Howdy! Welcome to our ENTO 423 final project overview.
Today, we will be taking you behind the scenes of our hypothetical infectious disease and its characteristics such as the vector, host, reservoir, agent, and our final control plan along with its implementation strategies.
First, we will start with the vector, Amblyomma maculatum, otherwise known as the Gulf Coast tick. This tick is primarily found along, you guessed it, the Gulf Coast region of the southern United States. It prefers warm, moist areas with high humidity and is the most active during the warmest seasons of late summer and early fall. It goes through three major life stages once hatched from its egg: the larval, nymphal, and adult stages. The larvae stay close to their hatching location until finding a desirable host, primarily small mammals like squirrels, which it will feed upon for 4 to 7 days, when it then drops off to molt into the nymph, taking about 1 to 3 weeks to do so. The nymph will feed on small mammalian hosts for 4.5 to 7 days, then takes 3 to 4 weeks to molt into the adult. The adult then finds a larger mammalian host, such as a human, and engorges for 5 to 18 days in the presence of males to mate with until it drops off to lay its eggs. It will lay an average of 8,000 eggs for up to 75 days. They are known to vector multiple diseases, including our disease pathogen, Clostridium cruciatus.
As mentioned previously, larval and nymphal ticks tend to feed on smaller mammals, such as squirrels, which just so happens to be our reservoir. The particular species at hand is the eastern gray squirrel, or Cirrus carolinensis. If you think I butchered this pronunciation, that's actually just how we say it in Texas. It is found in many terrestrial types, preferably woodland areas west of the Mississippi River on north to Canada, but has been found in other parts of the United States and the world. The peak activity for these furry little guys is during the spring and mating season and the fall, when they store acorns and pine nuts for winter. Because of their natural habitat, patients frequently encounter these creatures along the hiking trails and parks they have visited. Since it lives in the same habitat as the vector, it was easy for the larval and nymphal ticks to feed on them. All the squirrels had to do to encounter them was walk or run through vegetation where the ticks were questing, and attachment would be inevitable.
Now, moving on to the big issue, the human host. Let's start from the top. 10 different people were admitted to the hospital, tested for an unknown disease agent, and expired within a short time period. The major symptoms were high fever, fiery red epiglottis, tripod position, drooling, swollen or sore throat, dysphagia, and supraglottic structure appearing pale with watery edema. After some investigation, we determined that the patients who introduced this disease, our patient 0, were Frank and Patricia Howard. We believe they were infected while visiting the Bahamas on a cruise and brought the disease to the United States when they embarked at the ship port in Texas and further brought it home to Florida. Most patients had visited the same parks and hiking trails in Florida or had been near them, including Frank and Patricia. This is where the disease started to spread in a dangerous way. The map here shows the geographical range of the patients, the area they brought the disease back to after traveling.
Now that you know more about the background of the specific vector, reservoir, and human host, we're going to dive into the disease agent itself. Clostridium cruciatus is a dangerous, rod-shaped, gram-negative bacterium with a genome size of 950.4 MB. The early stage of this illness presents flu-like symptoms, with a characteristic swollen, fiery red epiglottis. Later on, as this infection progresses, the illness presents respiratory distress with low blood pressure and oxygen levels before the victim eventually falls into a coma and unfortunately succumbs to the illness. This disease collectively, with all its symptoms, is called respiratory Eastern disease, or RED.
The CDC currently has reports of 1,392 new cases with 200 confirmed deaths, and these numbers are expected to increase if action is not taken to control this disease. We have developed a control plan we hope will successfully decrease the number of new infectious disease cases in humans, as well as lower the numbers of positively tested squirrels and ticks. This plan will break the links between vector to host, pathogen to host, pathogen to reservoir, pathogen to vector, and vector to reservoir. We recognize that stopping this disease completely could be nearly impossible due to its ability to be maintained in the sylvatic and an urban disease cycle. Therefore, success for this plan is to find a significantly lowering number of new cases in humans and fewer numbers of positively tested squirrels and ticks. We will initiate this plan by first breaking the vector to host link. Avoiding tick bites is the simplest way to break this link, and there are several measures that will be taken to avoid these bites.
First and foremost, the parks where the vectors are present will be closing temporarily to the public until there's evidence of a decrease in positively tested ticks and squirrels. However, vaccinated hunters will be allowed in to hunt squirrels as part of the control plan. After reopening, wearing personal protective equipment such as long sleeve shirts, tucking pants into long socks or boots, and wearing insect repellent such as DEET will prevent tick bites and attachment. Other ways to avoid bites is to refrain from entering tick habitats in peak seasons and wearing light colored clothes so that ticks are visible and easy to remove. To implement this part of the plan and raise public awareness, signs will be posted in public parks saying that anyone visiting infected parks will need to be in the suggested protective clothing. We will send out all of this information in the mail to those within a 100-mile radius of these parks so that anyone who wishes to visit them will know how to dress before going. Any workers at these parks who see people coming into the park will also remind the visitors to take precautions against tick bites. After the signs have been posted for 2 weeks and the information has been sent in the mail, anyone entering the parks not in the appropriate clothing will get a warning, and on the second offense, they will be fined $50.
The next step in implementing the control plan will be breaking the pathogen to host link. In order to prevent this bacteria from causing disease in the human host upon exposure, we have developed a vaccine against the pathogen. Those vaccinated will be immune to the pathogen, so if they are bitten by a tick infected with Clostridium cruciatus, they will not get red. The only people required by law to be vaccinated are park personnel and hunters entering the park. For those who choose not to be vaccinated or are medically exempt, an antibiotic has been created for those who think they have been bitten by an Amblyomma maculatum tick and are not yet showing symptoms. Following the breaking of the previous 2 links, we aim to break the harder to reach links, pathogen to reservoir and vector to reservoir.
For breaking the pathogen to reservoir link, we plan to vaccinate the reservoir, the eastern gray squirrel, against the pathogen, Clostridium cruciatus, thus making it not susceptible to infection. We will implement this by conducting a catch and release program where scientists will set up bait and traps where the squirrel is known to inhabit so they can be captured. After capturing the squirrels, they will be vaccinated and released back into the environment. Another way to break this link is through using CRISPR on captured squirrels so that they are unable to become infected by Clostridium cruciatus. We can make it so that the trait is dominant and gets passed to the progeny of squirrels, which will be released back into the wild to mate. Future generations will then have the tolerance against Clostridium cruciatus infection. CRISPR can be used in such a way that it can genetically reprogram cells in the immune system to create an antibody without a vaccine having to be injected. This can be implemented with the same catch and release system mentioned previously.
To further decrease the prevalence of the pathogen, we will break the pathogen and vector link. This will be done indirectly by killing off the vector, but to ensure this link is broken, we will also kill the pathogen itself living inside the ticks. This will be done by spraying an antibiotic in an aerosol form into the air, on vegetation, and on the ground in parks and along trails that are most densely populated with positively tested ticks.
Finally, to break the final link, vector to reservoir, We plan on reducing both vector and reservoir through hunting seasons for the reservoir. This plan will include allowing hunters to kill the squirrels in the parks, raising the bag limit of squirrels, as well as extending the hunting season and offering a bounty for killing squirrels. Reducing the number of vectors also helps in breaking the link between the reservoir and vector. Since the pathogen cannot be passed to the vector from the squirrels to the ticks if there are no ticks for it to be passed. To reduce the number of ticks in the parks as well as other areas where the vector has been found, we have developed an insecticide that was made to target ticks.
Future outbreaks will be contained by closing off areas where the prevalence of disease are high and vaccinating the most at-risk populations. Through high surveillance, we will prevent outbreaks before they happen or catch them early when they do. The greatest issues expected to arise are with the public's cooperation in being vaccinated, not entering areas of potential infection, and taking precautionary measures like using insecticides and protective clothing. Consistently educating the public, advertising the risks of contracting the disease, the safety of vaccines, and enforcing current protocols will be very important in overcoming these issues as they surface. Another issue we may possibly encounter is the pathogen becoming resistant to the antibiotic. To prevent this, we plan to prevent the overusage of the antibiotic from the start. If there are any signs of resistance, we will develop a new antibiotic to fight the pathogen.
We expect full control to happen between 6 to 10 years. This time frame is dependent on the time it will take for hunting to significantly decrease the squirrel population. It is also dependent on how long the CRISPR gene editing takes to get mixed into the progeny population.
We hope to get to the point where people are no longer losing their loved ones to this horrible disease. A lot of families have dealt with loss, especially those living in the Florida area, and we want to do everything we can to put a stop to this devastation. If you are seeking more information, please visit our website at www.diseaseepidemiologygroup9.weebly.com. We hope you enjoyed our presentation. Thanks and gig 'em.